Boston. In this video, the science team and I are going to explain how we prepare and how we carry out our sampling out on the ocean. We begin by moving all of our equipment into the staging area. Next, we put on our float suits that keep us warm and safe while we're out on the ocean. Finally, we bring the boat over and load all of our instruments and equipment into the boat. Next, we head out to Station E to begin our sampling for the day.
The reason we prefer to sample at Station E is because it's the farthest from the shore and it allows us to collect water from up to 60 meters below the sea level.
When we go out into the field, we actually have two types of collections or samples that we make. One of them, we want to characterize what's happening in the physical environment. And some of what we do there is collect information about salinity and temperature and light from the surface down with depth. The instrument that we use for the salinity and temperature data is called the CTD. The CTD actually stands for conductivity, temperature, and depth. So by putting the CTD in the water and lowering down, we usually go to about 70 meters. We can then look at what the vertical profile is for temperature from the surface to 70 meters and the same for salinity. And we can see whether or not the water column is well mixed or if there might be some layers in there, because water of different temperatures and salinities makes different densities, and the different densities will actually separate from each other. The measurement of light is done with an instrument called a PUV. Is a profiling underwater sensor that measures both ultraviolet light as well as visible light, and that allows us to tell how deep in the water column phytoplankton are exposed to light from the sun. And of course light is required for photosynthesis, so it's important for us to know how deep that light reaches. And that depth can actually change quite a bit depending upon what's in the water column. So for example, the concentration of phytoplankton, as the number of cells increase, they're going to absorb and scatter light, so the light will not penetrate as deeply. If there are substances dissolved in the water, they will also absorb light, and that will have an impact on how deep the light can penetrate. So between the CTD and the PUV, we can have a nice characterization of what's happening with the physical aspects of the water column. The second part of the sampling that we do is actually collecting the biological material that we work with. So we will collect whole water samples, and this is done with the GoFlow bottles.
The team collects different whole seawater samples at 5 different depths: 5 meters, 10 meters, 20 meters, 40 meters, and 60 meters deep in the ocean. So this is a GoFlow bottle, and what's cool about this bottle is it allows us to collect water at very specific depths, for example 40 meters deep in the ocean, without collecting water from any of the other depths. So to do that, we send this whole bottle down on a line deep into the ocean, and it's got a hole in it so water passes through as it goes down. Once it gets to the depth that we need, we send a metal weight down that triggers it and closes it and captures, boom, whatever water is in it at the time that it hits it. Sorry, I got too excited. I shouldn't have said boom.
So that gives us what we call a whole water sample, and when that comes back to the lab...
So here we are, back in the lab after an exciting day of sampling.
We filter some of that water for chlorophyll concentrations. We save some of it for nutrient concentration analyses, and some of it gets filtered, and I look at which organisms are present so we can have a count of the cell densities for the different species that are found in the water. So in addition to the CTD and the bottle samples, the other samples we collect are in a plankton net. And what this allows us to do is concentrate the phytoplankton.
Hi there, I'm Joe, the scientist. We're out at Station E sampling today. That line you see in the water is a plankton net. Basically, we just drive around in a circle for 30 minutes and allow water to pass through the net and it collects phytoplankton. In a few minutes, you're going to see us pull the net up and see all the prizes inside. We'll return then.
So after the sample is collected in the cotton, the collected water is poured through a mesh to separate the larger zooplankton such as krill from diatoms. The diatoms are further concentrated and their RNAs preserved in order to stop degradation as well as any changes in the expression, so that later on when we are in laboratory in the USA, we can study what exactly was going on in the diatoms when they were collected in Antarctica. A part of the sample that is not preserved for RNA extraction is examined by DNAP to determine the relative abundance of species.
Of course, one of the things that's happening from the beginning of our sample season in August through to when we leave now in November is that we're going to be able to see a time series and see how these different parameters change as we go from the end of winter into spring.
Songs with the word ice in the title.
Ice, Ice Baby.
Yeah, Ice, Ice Baby is the only one I can remember because it came up the other day.
Yeah.
Ice in the title.
It's a tough one. Ice or snow?
Snow, snow. I know that.
Let It Snow.
From White Christmas. And that's the title. Snow, snow, snow.
Like there's gotta be, isn't there like an ice queen?